Good afternoon, Year 9. So, obviously we're going to be off school for a little while, so these video lessons are going to be continuing. Now, we're going to be doing a variety of different kinds of lessons, so some of them will be video lessons, some of them will be PowerPoint-based lessons, but we're going to continue on with the respiration topic. Now, on the boards, you will see, on your screen even, you will see the title at the top, and that written down. And the date, you need to write that next to it, you need to underline it. We're going to treat this exactly like a normal lesson. So by the end of the lesson, you should have a normal, fully laid out piece of work in your book. Now, the starter is a 6-mark question, and it's about pondweed, how you're going to use it to investigate its kind of life intensity on the rate of photosynthesis. Now, as I say, it's 6 marks. So in just a second, you need to pause me, read the question carefully, and write down the answer in full. And once you are done with that, unpause me and we'll go through the mark scheme together. So, please do pause me now. So, the mark scheme then. So the first thing to say is that this is not an exact mark scheme. In reality, any 6 mark will be levelled. And so you get 1 or 2 marks for doing a very basic kind of idea of how you do it, 3 or 4 marks for a method that would get you reasonable results, and 5 or 6 marks for kind of the perfect method. But the rough kind of things you need to be writing it. The general setup, you put the pondweed in the water, and you put the water in the beaker. And you shine the light on the beaker. So that is going to allow the pondweed to photosynthesize, because I hope we all remember that pondweed needs photosynthesis. Needs sunlight for photosynthesis, excuse me. So we're going to shine a light on the beaker and the pondweed will begin to photosynthesize. Then we want to measure how light intensity affects the rate of photosynthesis. And you're going to have to write down how you would do that. So you might move the light closer or further away, or you might do in some way turn the light up and down if you've got a dimmable light. And you're going to count how many bubbles of oxygen are given off by the pondweed in the time. Now, you also need to talk about how you control other variables. So things like, I'm going to put it in water in order to control the temperature so that it doesn't overheat. I'm going to keep the same piece of pondweed each time. I'm going to keep the same length of pondweed, same type of pondweed, same temperature of room, even that kind of thing. So you need to be writing down how you would control those variables. Leave sufficient time at each new light intensity before measurements taken. So this is about, once you put the light closer, you need to give the pondweed time before you measure how many bubbles are given off. And that's just to ensure that the pondweed has time to almost get used to the amount of light, so that it begins to give off the correct amount of oxygen bubbles for that amount of light. And then you're going to measure the rate of photosynthesis by dividing the amount of bubbles by the amount of time. So you're going to say bubbles per minute by the end of it. And finally, you always have to do repetition tests. Now if you write all of that together, will get you 6 marks. As long as your method would work, it'll get you 3 or 4 marks. And if your method wouldn't really work, i.e. you forgot to mention about controls completely, or you forgot to mention that you count the bubbles, you can't get more than 1 or 2 marks. It gives you an idea of the kind of question you'll see in terms of a 6 marker. So, today we're going to move on and talk about a number of different things. And in terms of learning objectives, we actually can't go through them right now, we'll be talking about them later. But the basic idea is we're going to talk about how gas exchange changes during the day and during the night. So, if I place a plant in darkness, i.e. I want it to be representing night time, so I'm not doing the experiment at night, I'm doing it in a dark room. Is carbon dioxide entering or leaving the leaves? That's the question. Is carbon dioxide entering or leaving the leaves? You can explain the reasoning behind your answer. So, give yourself 2 or 3 minutes. I want a full sentence answer. So it should begin, if a plant is placed in darkness to represent nighttime, carbon dioxide will blank the leaves, enter or leave, and then I want you to write a brief explanation explaining why you think that. Okay. So pause me for a couple of minutes. You need full sentences. I should not just see, enter the leaves. That is not a full sentence. Off you go. Second question. And we'll look at the answers. Now describe the levels of carbon dioxide leaving and entering a plant during daylight. So let's say I open up the curtains, shine the daylight into here. What would I see in terms of the amount of carbon dioxide leaving or entering the plant? And that's a little bit more complex than what's happening at night. You have to think about two different things. So again, pause me, write down what you would consider to be a reasonable answer, and then we will go through the answers in just a second. So full sentence answers. Fine. So by now you should have in your books an answer to question 1 in full sentence and answer to question 2 in full sentence. You should not just wait for me to give you the answers. That will be very obvious when your teacher checks your work later on. But let's do the answers now. So, correct anything you did wrong, basically. A single line through. There's nothing wrong with that, it just shows you've been thinking about it and you weren't quite right. Nothing wrong with that at all. So at night, no photosynthesis, that's the first thing. Because there's no light. Right? Photosynthesis needs light. Respiration is happening. So all living things respire all the time. Bacteria do it, plants do it, fungi do it, animals do it. Everything respires. That's why it's one of the Mrs. Gren things. Movement, respiration, sensitivity or senses, growth, reproduction, excretion and nutrition. First R, or the second, is respiration. So it's always respiring. Photosynthesis takes in CO2. Well, there isn't any photosynthesis, so no CO2 will be taken in. Respiration gives out CO2. Respiration is happening, so CO2 will be given out. So overall, we're not taking in any CO2, we are giving out some CO2, so overall CO2 will be given out. Now you may say to me, but sir, you told me that the stomata are closed at night. How do gases get out? And if you remembered that, fantastic. So the stomata, the holes in the bottom of the leaves that let gases in and out, close at night. But there are still tiny amounts of gases getting out just from the surface of the leaf, just from the cells on the surface. It's much slower, but it does happen. So we're talking about very small amounts of carbon dioxide being given out. Now in the daytime it's very different. Photosynthesis is happening. So there's light, so photosynthesis is happening. And because photosynthesis is happening, let me move my face so you can actually see the answer. We're going to take in CO2. Respiration is also happening, and therefore we're also going to give out CO2. So what's the balance there? Well, the answer is we do a lot more photosynthesis than we do respiration. We do a lot more photosynthesis than we do respiration because photosynthesis makes glucose. Some of that will be used for respiration, so it will immediately release the carbon dioxide again. But some of that glucose will be used for making cellulose for cell walls. That doesn't release carbon dioxide. Some of that glucose will be used for storage. Well, that doesn't release carbon dioxide. And so overall, we're going to take in a lot more carbon dioxide. We're going to make a lot more glucose than we're going to give out carbon dioxide. We're going to release glucose. All right. So overall, in daytime, we're going to give out. We're going to take in CO2. Excuse me. We're going to take in CO2. We're going to give out oxygen. Fine. All right. So let's keep going now. Why do I ask about these questions? What's the point of these questions? The point is we're going to talk about this indicator called hydrogen carbonate indicator. And it changes colour based on the amount of carbon dioxide present. So have a look at that, and I actually want you to copy this down. I want you to write the words hydrogen carbonate indicator, and I want you to write down the sentence and what colour it goes. Now remember, well, I haven't told you yet, but you should remember, no one's ever going to ask you what colour hydrogen carbonate indicator will go. The question is not going to say, what colour does hydrogen carbonate indicator go when CO2 levels are high? But they give you this information and expect you to understand it and apply it, as we're about to. So I'd like you to write this down just so when we ask some questions about it in a second, you will know what colour it will go. So CO2 levels, when they're low, it turns purple. When CO2 levels are normal-ish, it stays about orange. When CO2 levels are high, it goes yellow. So pause me if you need to, copy down the blue sentence and the diagram at the bottom, if you want to call it a diagram. Right. So, now, we're going to design our own experiment here. I'm going to do this experiment in school when you come back. It's one of the experiments we like to do. You're going to be given any simple equipment around the lab, so we've got all kinds of stuff around the lab. Hopefully you can picture most of it. Have any equipment from the lab. We're going to give you some plants. We're going to give you maybe some small animals like a mollusk. And we're going to set it up to find out what happens to a plant during the night and during the day. So you need to design an experiment that would enable you to find that out. And hopefully what you should find out is what we just talked about. But you might not, you might find something different, in which case we've just disproved one of the laws of science, that's fine. Right, so, two things to say. Firstly, if you're going to place animals in, animals and not plants, you're going to store them in a little wire mesh kind of a net, almost, so that they don't go into the indicator. So you might put the indicator at the bottom of, say, a beaker or a test tube, and then you're going to put the animals in a little wire mesh kind of hanging above that, so that they can't fall into the indicator and die, which would obviously be unpleasant for them and for you when you have to clean it up. Now, you're also going to treat the animals ethically, so you're not going to mistreat them or put them in a position where they're going to be in pain or are going to suffer in any way. So we're going to think about that if we're going to use the animals. But you're going to set up an experiment where you've got the indicator in a place where the plants are also there and the animals are also there in places. You're going to find out how changing the night, changing the day is going to affect that. So what I'd like to see in your books written down, and this should take a little bit of time, is a method written down explaining what are you going to do with these different pieces of equipment. Now, the only final thing to say is that hydrogencarbonate indicator is not harmful, so it can be placed in a beaker. Well, I take it back, it is harmful, but it's not really noxious gases or anything like that, and so it's going to be placed in the bottom of a beaker or in the bottom of a test tube quite happily. It can be left for quite a long period of time. So you might want to do that. So pause me, have a think about, right, I've got this indicator, I need this to end up in the same place as a plant, in the night and in the day. Maybe I'll include some animals in somewhere. How am I going to do this? How am I going to set this up? What would it look like? You're going to write yourself a little method, maybe draw yourself a little diagram. And then we'll talk through what you might end up doing and add to your method. So pause me for 5 or 6 minutes, it will take you. So, hopefully you've had a go, you've got yourself a method. Now this is difficult. I'm going to show you the kind of thing I'm thinking of. You can actually do something subtly different, but here's the kind of thing I'm thinking of. So, here are the things that people would usually set up. So we've got one without anything in it. So here's a boiling tube with this nice indicator. You can see it's orange to start with. This nice indicator. One with a bung in the top, but nothing in it. One with a bung in the top and a leaf in it. One with a bung in the top and a leaf in the dark. One with a bung in the top, and there are some snails in here. I take it back. There are no snails in here, just the wire mesh. 6 has the snails in it. So we've got nothing with a bung with a leaf with a black thing around it, bung with a wire mesh and nothing, bung with snails in a wire mesh. And you can try different kinds of things. You can try putting the peas in there. See if peas are different to leaves. You can try putting the leaf in the indicator and see if it makes a difference. Try putting the leaf in the indicator in the dark. So all of these things are set up. Now, what I'd like to see in your books is a labelled diagram of this equipment. And it should look like 9 test tubes lined up with what is in inside each one, drawn and labelled. Now, that's very straightforward, that first bit. Underneath that, for each test tube, you should write why does it exist. So you should label what's in it, and then underneath you should write, well, in test tube 1, that exists for this reason. What is the point of having an empty test tube with just indicator? What is the point of having an empty test tube with just indicator and a bung? Why do those exist? I think we can all understand why I need number 3. I want to see what happens if I put a leaf in. I want to see what happens to the amount of carbon dioxide produced. I'm hoping that carbon dioxide should go down because it's going to take in carbon dioxide, but we'll see. Similarly with the dark, why have I put a leaf in the dark? And so on and so on and so on. So you should have a diagram of 9 test tubes labelled with what's in each one, And underneath you should have written down why each test tube is used. Why have I included it? Fine. Now, after all that, you need to then have completed all the tasks for this lesson. You need to upload that to Teams for your teacher. And that should be the end of this lesson. I imagine this next task will take you about 8 or 9 minutes, this drawing and writing task. And once you're done, it's the end of the lesson. And next time in school, we'll be completing this practical. Okay. Thank you, and I hope this goes well for you.